Morning, Simon. Where are you at the moment? You don't look like you're in your office, or it's changed.
No, I'm at home in Yarnton. I got back from a conference in Los Angeles and unfortunately got the lurgy almost immediately. And
Oh,
so struggling to be back in at work at the moment.
well, I hope you're on the mend. What was the conference about?
It was the Biophysical Society's big annual meeting. It's something, I counted this year. This is my 19th year going to that meeting.
You
So
probably know how it works.
yeah, you'd have thought so by now. It's a great, huge meeting. Lots of biophysicists from around the world. And great to catch up with colleagues, friends, and see some mind-blowing science, to be honest.
Well, and I hope you've been paddling the podcast and finding some future guests.
Absolutely. There's a lot of interesting stuff going on, especially with artificial intelligence playing more and more a bigger role in biology. I think there's exciting times ahead.
Excellent. Well, today I'm quite excited by today. We're one of the things that's been in the news recently, or the scientific news at least. There's a couple of papers published recently where this spacecraft called OSIRIS-REx left Earth. It flew to a asteroid. It went around called Bennu. It turns out this asteroid, was basically a pile of rubble turning out through space. They then sent down this kind of this little bit of equipment that grabbed a load of material from this ball of rubble, took it back to the kind of this is I'm probably got this completely wrong. Took it back to the main spacecraft, then brought it back to Earth, dropped it off on Earth, and now this spacecraft has cleared off somewhere else. Anyway, the first person we're going to talk to has been involved in this amazing mission. She works at the Natural History Museum. She's called Sara Russell, and I'm really looking forward to hearing about that mission because they found some really interesting stuff.
Yeah, and I think the exciting thing here is they found it and captured it in a completely uncontaminated way. So for the one of the very first times, we have direct material, which has been untouched by human hands, as it were.
And I think they've found some quite important chemicals in it, haven't they?
One or two that are fairly important, it seems.
Yeah. Well, we'll be hearing about that
Yeah.
from Sarah. And then we'll have a chat with a chemist who's actually thought quite a lot about biology and about the chemistry biology interface.
Yeah. So Addy Pross from Israel is going to tell us about well, how life may have emerged from chemistry.
I know. So by the end of this podcast, we might have some understanding of why we're here and what we're doing.
Sure, and hopefully our listeners will. Whether you and I will, Tim, I'm not so sure,
Well,
but
yeah, I
the
don't
others.
think we really know what we're doing yet. Anyway, let's get on with it. So we will. Uh, Sarah will be joining momentarily, and we will be chatting with her. It's our great pleasure to have Sara Russell from the Natural History Museum in London join us today. Sarah, perhaps you'd like to introduce yourself.
Sure, Tim. So my name is Sara Russell, and I am a researcher in planetary sciences here at the Natural History Museum in London.
Okay, great, Sarah. So we're absolutely been reading about the great discoveries of this wonderful spacecraft, OSIRIS-REx, and obviously you've been giving lots of talks and been incredibly busy recently. We're delighted you're
Yeah,
here with us today. Can you tell
thank
us
you.
tell us about OSIRIS-REx and what it's found?
Yeah, so OSIRIS-REx is a NASA space mission, and it's been a very long time in the planning, but it launched in 2016 from Cape Canaveral, and it went to visit an asteroid called Bennu. And Bennu was targeted because it's what's called a C-type asteroid, and C stands for carbon. It's a very dark asteroid, and we were hoping the fact it was dark would mean that it's rich in carbon and water. And so this was chosen as the target because one of the main aims of OSIRIS-REx was to learn about our origins, to look at asteroids that may have been building blocks of planet formation that may have provided all of the carbon and water that, for example, the Earth took and allowed it to become habitable. So it spent 2 years looking at asteroid Bennu. It's about a 500-meter across asteroid. And then in the most kind of exciting stroke, terrifying part of the mission, it actually sampled the surface of asteroid Bennu. So it had an arm that came down with a sampler on the end of it called TAGSAM, or touch and go sampler, and it just touched the surface for a few seconds, and that enabled the sand and rubble that was on the surface of Bennu to be captured and ultimately brought back to Earth. So it came back to Earth in September 2023, and we've been busy analysing it ever since.
Oh, wow. So you've had access to the exact samples that came from the asteroid?
Yes, so the samples were sent to laboratories across the world. There were over 200 scientists involved in the analysis, and we were really lucky to get a portion of it at the Natural History Museum to analyse.
And how did you get involved? So Natural History Museum, you know, I think of it as dinosaurs and what have you, of course, but
Tim. Yeah. No, we have a great dinosaur collection, but yeah, and maybe this is something people don't know about the Natural History Museum, but one of the other collections we have is our meteorite collection. We've actually been collecting meteorites for over 200 years, and so we have all of this experience in space rocks and what they're made of, and particularly most meteorites, so meteorite is any extraterrestrial object that's fallen to Earth naturally. Most of them come from asteroids. Most of them come from near-Earth asteroids. So this kind of gives us a particular expertise in knowing what asteroid Bennu may have been made from. And yeah, so in the basement of the museum, we have most of our collections, and we also have a lab facility so the scientists can work on our collections behind the scenes.
And how did, and so you got obviously you're an expert in asteroids, but also the moon and the Earth and what have you. So did you get an email out of when this was all starting off from NASA saying
Yeah.
you know, how do you get involved in these things?
You know, it's been something that has been in the mill for decades, honestly. And I remember as a PhD student, which is like a really long time ago now, just talking to my fellow students, and one of the problems, so meteorites are fantastic objects to study and so interesting, but one of the problems with meteorites is that they land on the surface of the Earth and you don't know where it's come from. So, you know, we had to figure out, it took a long time to figure out that most of them did come from asteroids. So there used to be lots of theories that they used to all came from the moon, or they all came from comets or the sun, whatever. But anyway, we figured out they do come from asteroids, but it's very hard to pinpoint exactly which one. So we don't have that context. So me and many of my meteorologists colleagues have a background in geology, and we're taught on our first geology lecture that we have to go out into the field to learn about, you know, where our samples come from before we can actually analyze them and understand them by looking at them in the lab. And so OSIRIS-REx is like our meteorologist's big field trip. So we went out to explore an asteroid. We, you know, instead of having a field assistant, we have a robot who's doing all the work for us, but bringing the sample back for us to study. And actually, it's sort of my generation that's kind of leading the mission. So, you know, I've known them all for ages, basically, and there is kind of feeling that we all did this together, which has been really super fun.
And so, can you tell us, have there been surprises in your findings?
Yeah, there have been loads of surprises. So, and it makes us realize, you know, how much we didn't know. So the big surprise, so we do have a class of meteorites called carbonaceous chondrites. So these are ones that also look dark. And we thought these would probably be similar to asteroid Bennu. That's what we were expecting. But there's quite a wide variety of types of carbonaceous chondrite. And the first thing we realized about Bennu is that it's most similar to a type of meteorite called CI chondrite. And this is one of the very rarest types of meteorite. There's only 5 observed falls, so out of 70,000 known meteorites. So it's incredibly rare. And the other weird thing is that there's actually been 3 asteroid sample return missions. Two of them were by Japanese missions called Hayabusa and Hayabusa 2. And of those, 2/3 of the asteroids returned look like these CI meteorites. So another thing about these CI meteorites is that they're very delicate, they're very fragile, they've been incredibly altered by water. So they're basically just like little lumps of clay that break apart really easily. So I think what that's telling us is that these are really common things out in space, but we don't get to analyze them very often as meteorites just because of the bias of things coming through our atmosphere and managing to survive this kind of very traumatic event of landing on the surface of the Earth. So it's telling us about our meteorite collection as well and how that might be biased. So that was I'll just carry on. So that was the first surprise. And then the other kind of big surprise as we delved into it in more detail was we found all of these mineral salts inside Bennu. So these included sodium chloride, table salt, also potassium chloride. We also found sodium carbonate, which is another like really soluble minerals. So we found all of these things that disappear really easily in the presence of water. And what we think is that these salts formed from a briny pod of water. So this tells us there was actually free water on Bennu, which is not what we expected. We thought that Bennu probably accreted with ice. So ice and rock accreted together. And then the ice would have melted, but just infiltrated into each individual rock rather than being a big body of water, which is what we need to form these salts. So then these pods evaporated away and left behind these, what we call evaporite deposits.
And so that evaporated, so it formed as ice and then it melted as it came closer to the sun. So what's the sort of trajectory of Bennu look like? Has that been worked out as well?
Yeah, a little bit. So we think that Bennu formed originally right in the outermost part of the solar system. So there would have been loads of ice and also things like ammonia, which is also very rich in Bennu. So in these very, very cold outer regions beyond Jupiter, maybe even beyond Saturn, like way out in the outermost part of the solar system. And then the ice would have melted, maybe not melted because it came close to the sun, but melted because it accreted also with radioactive isotopes that would cause some heating of the body to start with. But at some point, it did get ejected first into the asteroid belt between Mars and Jupiter. And that may have happened very early on in solar system history. So we think that the giant planets actually migrated around when they were forming. They may have come into the inner solar system, then out again. And this movement basically disrupted all of the smaller bodies like Bennu and other asteroids and comets, and forced some of them out of the solar system completely, but some of them kind of got to jump into the innermost part of the solar system. So it's really kind of telling us a story that maybe we didn't appreciate so much before, that things that move around so much kind of radially in their distance from the sun, as well as orbiting around the sun.
So isn't that one of the reasons that Mars is thought to be small, because Jupiter did this grand tack thing and started going in towards the sun, and thank God it got pulled out again by Saturn, otherwise we might have been gobbled up.
Yes, exactly. So there used to be this big mystery about why Mars is this titchy little runty planet, whereas you would have thought it would have been formed in the middle of the disk. It should be quite a substantial planet. But yeah, we think that's because Jupiter kind of blasted its way into that kind of region and sucked up a lot of the material that should have been part of Mars.
No, it's really fascinating. And so where did the interception happen? How far from Earth was it that the OSIRIS-REx mission did its touch and go? And was it touch and go? You must have been really nervous when that was happening.
Yeah. I mean, the distance between Earth and Bennu is continuously changing, because although Bennu has a Earth-like orbit, it's sometimes on the other side of the sun and sometimes nearer to the Earth. So it comes closer and further away. But the touch and go was completely terrifying. So we always knew that would be the kind of scariest part of the mission, because although we'd spent 2 years kind of studying Bennu, the actual collection of the sample would only take 5 seconds, so kind of 5 seconds of absolute terror. And it was even more terrifying than we expected, because when we were planning the mission and we did our tests for the sampler, we were assuming that it would be a fairly kind of solid surface, like something you could walk over, but actually the surface of Bennu is a rubble pile, so it's just like this pile of rocks that are very kind of loosely touching each other, just gently held together by the very, very small amount of gravity. And the sampling arm kind of just went through the surface there, like as if it was going through a ball pit. And it's only thanks to the fact there are little thrusters on it that's let it jump back. Otherwise, the spacecraft would have just sunk through the whole asteroid.
And were you monitoring this as it happened live?
Yeah. We were watching it for sure. We were watching it online. Yeah. And it was horrible. It was horrible. I mean, there were 3 terrifying times. There was the launch, there was the touch and go, and return to Earth, which was also pretty nerve-wracking as well. But thankfully, everything worked, which is miraculous, really.
Amazing.
Well, amazing. I mean, it's a testament to Thank you. Well, it's testament to the brilliant engineers that worked on this that made it all work.
And am I right that the samples were dropped off on Earth and the spacecraft continued off somewhere else?
Yes, yeah. It's sort of like a Deliveroo driver or something. So the spacecraft just came to Earth and dropped the samples at the top of the atmosphere. And actually, because the spacecraft is still working fine and all the instruments on it are still working, it was decided to extend the mission. So now OSIRIS is called OSIRIS-Apex, and it doesn't have the capacity to collect any more asteroidal material, but it's going around and surveying near Earth asteroids. So the mission is actually continuing, which is really exciting. But the space capsule, the return capsule, was dropped at the top of the atmosphere, and then it fell down to the Utah desert. It had a parachute on board. So one of the things we were worried about was whether the parachute would open. So there have been 3 NASA missions that have used this technology. There was the Stardust mission that went to a comet, Genesis mission that collected some solar wind, and then OSIRIS-REx. And of those, the Genesis mission actually crashed. So the parachute didn't open properly. So we were worried, because it was a similar technology that was going to be used for OSIRIS-REx, but it opened all fine. Everything worked fine. So
thankfully.
All good. Thankfully, yes.
So can we just go back to the findings of the sample? So you've talked about the minerals and the salts that have been found, but excitingly, you also found some bigger building blocks of life. Can you talk us through those a little bit, please?
Yeah, sure. So I'm not on the organics team, but it's actually brilliantly the mineralogy team and the organics team are finding something that's a complementary story. So if we go back to these pods of water on Bennu, so we think these would have been salty, alkaline, briny pods of water. And one of the exciting things about having brines, well, actually, it's 2 exciting things. One exciting thing is we think that there are brines across the solar system. So we think the moon of Saturn, Enceladus, for example, is spewing out briny water, and we see salt deposits from brines on Ceres. So although brines and the evaporite deposits are kind of minor things that happen on Earth, they might be really widespread things across the solar system. So we're really lucky to have these samples on Earth to be able to study these processes. We're really excited that we're potentially seeing a widespread process. But the other exciting thing is that brines are great places to make organic molecules. And our organics colleagues found these really complex and life-important molecules such as amino acids. So they found 15 out of the 20 amino acids that are used in life. And they also found all 5 of the nuclear bases that are used in life, the 4 that are used in DNA and uracil from RNA. And what we think is that probably Bennu accreted with these ice and the rock, but also very simple organic molecules, which then kind of cooked up in this brine and produced these much more sophisticated organic molecules. And as well as these organic molecules, this array of salts are also really important to produce life. So, for example, one of the salts that we also weren't expecting, because we don't see them in meteorites, was phosphates. We see a lot of phosphate minerals, including a sodium magnesium phosphate, which we weren't expecting at all. And of course, phosphate is also really important in producing DNA and producing life. So we seem to have all of these life ingredients kind of mixed together in Bennu. In Bennu, life didn't start, and then so the question is why not? But still, it would have delivered. Asteroids like Bennu would have delivered all the ingredients to the Earth, but also to Mars, everywhere across the solar system. And maybe the same process happens on exoplanets around other stars as well, because these, I don't think there's anything particularly unusual about Bennu. So it's really exciting that we might be kind of trying to glimpse into how the origins of life happened.
So congratulations on your paper that came out, an evaporative sequence from ancient brine recorded in Bennu samples. That was just a couple of weeks ago in Nature. That's always fantastic to get a high-profile paper. It's
I know, it was great.
Yeah, we'll put a link to it in the in the chat. But
Thank
now
you.
I'm going to go a bit science fiction-y here. So could life start on an asteroid like Bennu, do you think? And could you have ended up bringing like a killer bacteria back to Earth to take us all down? Were you playing an incredibly risky game by
I don't think so. So every space mission before it gets approved has to be assessed for planetary protection. So you have to show that you're not going to be bringing back killer alien bugs to Earth. And that's one of the reasons, for example, the Mars sample return program is so complicated, because Mars has been assessed as a site where there might be still living life. And if that's the case, then we don't want to bring back these weird Martian bugs that may interact with the Earth's biosphere in ways that we can't predict. But asteroids, thankfully, have got a status called unrestricted, which means that it's assumed that they are sterile. And part of the reason for that is because we have this massive meteorite collection on Earth, and there's no evidence that any of these asteroids that have fallen naturally to Earth have got any live bugs in them. So hopefully that won't happen. And it's probably because asteroids are small bodies and they're airless bodies. They're being continuously radiated by radiation from the sun and from the galaxy as well. And that will have a sterilizing effect on them.
Well, that's good to hear. Although I hate to imagine what your health and safety forms look like.
I guess there's still a huge leap between finding those building blocks that you found on Bennu and then life. So we should be safe.
Do you want to speculate?
For sure.
Do you want to speculate how life got started?
I have no idea how life got started. I wish I knew. I'd be collecting my Nobel Prize, wouldn't I? Yeah, I don't know. It's sort of frustrating that we sort of have these building blocks and we have fertilizer on Bennu in the form of phosphorus and potassium and nitrogen, and we have water, and we have these really kind of sophisticated organic molecules. So yeah, why didn't it start, and how does it start? It's complete. It's a mystery to me anyway.
Yeah,
maybe it will in the future. Bennu's still going, so
Bennu's still out there. Yeah.
I mean, I guess it's possible that something starts there and evolves in a slightly different way to, it looks a bit different to life as we have it on Earth. It's still possible, I think. I would imagine. Do you think? Yeah,
I mean, now that Bennu's so close to Earth, it means it's also so close to the sun, and it's being radiated more. So it has, it's in the Goldilocks zone of having being in the right sort of temperature environment, but it does have quite a hefty radiation dose where it is. So I would imagine that it's not going to start there.
No, well, it's really exciting work, and what a great time to be, working in this area when you're able to get, off-planet samples returned and all the exciting things. And I'm sure we're going to hear about
Oh, absolutely.
many more exciting findings from Bennu, because it's just the first few papers are coming out at the moment. So
Yes, exactly. It's just the first few papers. We've still got a lot. There's a lot in the pipeline. But also, yeah, I think you're right. This is maybe kind of the beginning of a new era. So, I mean, you said it was odd that I worked at a natural history museum, but maybe this is what natural history museums are going to do in the future. They're going to explore the solar system and fill our museum with collections from all over the place.
And I must admit, I did know that you had a meteorite collection, but I thought
I know you did.
So Sarah, can I just ask you? You touched upon that OSIRIS now no longer has the capacity to collect samples, but it's still monitoring. What sort of signals what sort of information is it sending back now?
Yeah, so it has a fantastic camera, so it can take pictures of asteroids. It also has a spectrometer on board, which can give you an idea of the composition of the different asteroids that it's studying, and a LiDAR, so it can make shape models. So, and then we can make 3D models of them on Earth. So it finds out about their composition, shape, what they look like, that sort of thing. So really, yeah, a lot of really interesting information. And of course, it's really interesting to look at asteroids that are near Earth as well. It means we can better monitor potential hazards to the Earth in the future.
Well, thank you very much for joining us, Sarah. I really appreciate you spending some time talking to us. And really
absolute pleasure.
Really exciting work, and great to hear it. We're going to actually talk with someone who's been thinking about how chemistry becomes biology next. So it'll be interesting.
Great, they can tell me.
Yeah, so, no, I don't know. We'll hear what he says. But good luck with the rest of the analyses from the samples that have been brought back and all your other work. It really is fascinating stuff. And thanks for taking the time.
Yeah.
Oh, thank you very much, Tim and Simon. Thanks.
Thanks a lot, Sarah. Today, it's a great pleasure to have with us our guest, Professor Addy Pross from Ben Gurion University in Israel. So Addy's been a professor of chemistry for many years, and his particular interest is really at the interface of chemistry and biology. He has a wonderful new book out, which is called What is Life? How Chemistry Becomes Biology. And we're hoping to probe him a little bit more about those very questions today. So welcome, Addy.
Thank you. Great to be here.
So one of the things that's been in the news a little bit recently, Addy, is this remarkable spacecraft that flew over to an asteroid called Bennu and picked up a load of dirt and a load of samples and brought it back to Earth. And the first results are just coming out from that. And this has confirmed previous work that shows that a lot of the key building blocks of life are actually made off planet. They're made in space. And I suppose that, the kind of key first question is, does this mean that we're going to find life on endless other planets, do you think?
Well, that's, of course, a tough question, but I don't think it depends so much on the kind of molecules we find. I think in order to understand the life phenomenon, the life process, we have to understand how that kind of process can get going. And that doesn't have to depend that explicitly on the particular molecules that one uses. The point is, if you go to the best chemist in the world and say, listen, I'll give you whatever materials you want, proteins, nucleic acids, whatever you want, in any quantity you want, under any conditions that you would like to employ, can you make me just a very simple little living thing? And the answer will be, well, they don't know how to go about it because we do not understand the process yet. We do not understand the process by which dead stuff becomes alive. So my focus as a chemist is to try and understand that process. And to put it bluntly, physics and chemistry have left you biologists in the lurch. We haven't helped you understand what life is, and you biologists have been forced to look after yourselves, to just do it, to think about the problem in a biological way. but with all due respect, the problem goes deeper than biology. The roots have to be in physics and chemistry, and that's what I'm struggling with.
But Addy, is it sort of, does it come as a surprise to you that it would be these very same amino acids and these precisely the same 5 bases in the exact same chemistry we have on this Earth, which is out there also in space? What are the chances of that?
Look, that's a good question. First of all, I should say quite frankly up front that I'm not familiar with the details of the findings of that particular expedition. But as I say, you may be right that there is some relevance there, because clearly, when I say the process doesn't depend on the particular molecules, that's not entirely true. Clearly, some molecules will be more susceptible to undergoing the necessary process, and we may, as we get on, talk about that. And a key feature for that process has to be replication. Replication is central to the emergence of life, but it's not sufficient for replication to begin with a molecule that has the characteristics of being able to replicate. It's already almost 70 years since a very clever guy in America, Sol Spiegelman, was able to show that if you take an RNA molecule, you can make it undergo multiple processes of replication to make copies of itself by feeding it its building blocks. But we've been working with replicating molecules for 70 years, and that hasn't got us anywhere closer to understanding what life is, because when he started out with a 4,000 long string, RNA string, it replicated, occasionally mutated, but it mutated in the direction of becoming shorter, because shorter strands were faster at replicating than the longer ones. And therefore, it was evolving, if you like, away from life and not towards life. So we've been stuck really at the point that a replicating entity is not the solution in itself. So now, there has been, I'll just say one comment, and we won't take it further at this moment. There has been a dramatic development in systems chemistry from about a little over 10 years ago, which changes the whole story, and we'll get onto that soon. A new kinetic state of matter has been discovered, and that is a big deal.
Well, let's hear what that. So tell us about that, and then I'm going to come on and ask you what is life. But tell us about this. Tell us about this exciting kinetic new state that was discovered about a decade ago, and which you cover in your book, of course.
Yeah. Well, of course, everyone with some physical and chemical background knows that there are the thermodynamic states of matter: solid, liquids, and gases. But two Dutch chemists in 2010, Jan van Esch and Rink Ilkema, discovered that one can do a very common reaction in organic chemistry, esterification, but instead of just making the ester product in the normal thermodynamic way, they managed to put it into this dynamic kinetic state way. In other words, they created a dynamic system in which the product was very different to the thermodynamic product that you do in the normal way. Now, that sounds confusing. So just to give you an idea of what that is, a physical analogy might be helpful. A very simple physical analogy, a water fountain. Now, a water fountain is stable in the sense that as long as the water is provided and as long as there is a pump to push the water up, you have this fountain, which is not thermodynamically stable, it is kinetically stable. It's non-equilibrium, but it's stable as long as energy is being pumped in. And And the other interesting thing about this water fountain is that the water that makes up the fountain is turning over all the time. The fountain is supposedly the same fountain. There's this wonderful fountain in Geneva. Whenever you go to Geneva, there it is doing what fountains do. But the water is continually turning over. Now, in this dynamic kinetic state that was just discovered, it's the same situation. The material has certain properties which are very different to the thermodynamic properties, but the molecules within that state are continually turning over. And the point is that a new dimension of chemical possibilities was discovered, a kinetic dimension, a dynamic kinetic dimension. And life is basically, if we want to categorize it in physical chemical terms, it's just some material that has accessed that new kinetic dimension. So both of you, as we speak here, are continually turning over. In 2 months' time, all the stuff that's you is gone. And basically you are new stuff, but of course it's still you, just as the fountain is still the same fountain.
So, Addy, in a way, you're saying that you use an energy source to keep us far away from equilibrium.
That's correct.
Okay, because if we reached equilibrium, we would just stop.
We wouldn't stop, we would die.
Yeah,
That's why we die once that dynamic state is terminated. And we can do that to a chemical system, of course, then it is the equivalent of dying.
Yes.
So how do you switch the waterfall on, in the water fountain on in the first place? So presumably, I mean, that doesn't come from nowhere. That must come from a mix of chemicals or, a particular set of environmental conditions in order to get to get the water fountain flowing.
That's true. Because life is dependent on continual supply of energy, and we get that energy through munching, through lunch and dinner and so on. But initially, it appears that the earliest life forms used photosynthetic energy. Earliest bacteria were photosynthetic. So how exactly that happened and what happened, we don't know. But my point is that if we want to understand the life process, and in particular, and this is very important, the evolutionary process, where does evolution have a direction? There's a lot of confusion about what evolution is about. But once one understands that it is matter in this dynamic kinetic state, then you have a handle on what the evolutionary process is directed to. And the answer is, when I say life is in the dynamic kinetic state, that's not sufficient. It has to be replicative as well. In other words, replicative and in this dynamic non-equilibrium state. Now, I have to tell you, for chemists to make something like that is very difficult, and there aren't too many that have made too much progress there. But the beginnings of some progress are there. There's one systems chemist in Groningen, Sibrin Otto, who has shown that there are indications that once you have such a system, it tends to move in the direction, if it undergoes a mutation or change, it moves in the direction of becoming more kinetically stable. In other words, there is a direction of change from less stable in this DKS sense, dynamic kinetic stability sense, to more dynamic kinetically stable. And incidentally, that process of increasing stability comes about through a process of complexification. In other words, once that system acquires, let's call it, more knowledge about its environment, because you see, a DKS, that's the difference between a thermodynamic state and a DKS state. That decay state is in constant communication with its environment because it's totally dependent on its environment. If the supply of matter or energy is stopped, we already said death. So it's learning constantly about its environment and incorporating into itself whatever features will facilitate its staying stable, persisting in that environment. Now, there's maths in there as well. Just as the maths of the second law show that matter tends to go from less persistent to more persistent, or in statistical terms, less probable to more probable, the maths of dynamic kinetically replicating systems says that better replicators will drive poorer replicators into extinction. The math says that. So basically we have here, nature has discovered two ways of moving towards persistent forms. The traditional thermodynamic way towards higher entropy and lower energy, where the mathematical directive is one of probabilities. And then nature discovered this other way of reaching persistent forms, and this is through a dynamic kinetically stable system becoming more complex to become more and more persistent.
So trying to kind of put that in a slightly different way. Once this kinetic dynamic state starts, so this kind of new way that matter's behaving starts, it's inevitably going to move further away from from the equilibrium of its surroundings. Is that what it's going to get more complicated, and so the sort of distance, as it were, between the replicating dynamically kinetic being is further away. It gets ever further away from the environment around it, but it depends upon that environment and it modifies that environment.
Yeah, I would say from the first moment, it is in a non-equilibrium state maintained through energy supply, just like that water fountain. So from day one, it's non-equilibrium. However, even though it's unstable thermodynamically, it has it persists as long as energy and matter are supplied to maintain that state. But if that state can evolve in a way that makes it, let's say, less susceptible to problems like losing the source of energy, losing the source of material, then it will complexify in order to maintain its state in a better fashion. So clearly that first state didn't have a membrane, for example, and then the system discovered that if you have a membrane, then to protect itself from its environment, but to allow stuff in and stuff out, which is essential for survival, then that is a more persistent, more stable in this dynamic kinetic sense. So there was a general tendency to move towards more complexity, if you like, learning from the environment what is useful to persist in that environment.
So, Addy, as a chemist, I think when I'm listening to you, in my mind there's still a disconnect between, okay, we have this beginnings now of a system that can replicate and it's learning from its environment. However, for it to really thrive, it will need at some point sophisticated chemistry that also has to be done in a kinetically viable fashion. So at some point we need enzymes, we need to lower energetic barriers. And that still seems to me to be a leap between what we have now, what we're discussing here, and this other chemistry, the one that needs, to make a covalent bond is perhaps the hardest thing. And so, can you talk us a little bit through that?
Well, look, what you said is absolutely correct. This movement towards greater complexity initially will come about through creating reaction networks. That's the beginning of it. So we all understand that cooperation facilitates biological function. That applies in the DKS sense as well. Once you have several reactions in a network, that may well be more stable in this dynamic kinetic sense, because let's put it this way, a greater ability to respond to perturbations. Maybe, I don't know if it's of interest, you have to tell me. I can tell you the system that Vanesh and Ilkema made. They made a hydrogel in this system, which is very different to what you get when you do the reaction thermodynamically. But the beauty of that hydrogel was that by playing with the kinetic parameters, you could change its qualities. You could effectively, and this is remarkable, above any thermodynamic state, there are potentially an infinite number of dynamic kinetic states, because you can play around with the kinetics to end up with that dynamic state in many, many different ways. So if you want the hydrogel to be a more rigid one, you change the reaction the rates of the formation of the materials in one way. If you want it less rigid, in another way. That dynamic capability is what gives life its extraordinary functionality. It plays around. If it's got thousands of states of similar DKS, dynamic kinetic energy, it can shift around between one and the other, and it's doing that all the time. And the best example of that, by the way, is the cytoskeleton. The cytoskeleton, we all have skeletons, but that cytoskeleton, actually like our own skeleton, is in a dynamic kinetic state. In other words, you have there a situation where microtubules are formed reversibly with tubulin dimers, and the rigidity of the cytoskeleton depends on the amount of the two materials there, microtubules as opposed to the dimers that make up that microtubules in a polymerization reaction.
So just for our listeners, the cytoskeleton, you're talking about what's going on within cells here, aren't you?
That's correct.
Yeah. Just
That's
in case
correct.
Our listeners are not sure of the phrase cytoskeleton.
That cytoskeleton, and in fact, many of the organelles within the cell are in this DKS state, constantly turning over. So the cell itself is in this DKS state, turning over. You know, in your body, many of your cells are disappearing continually, skin cells in particular, blood cells. But within the cell, organelles are being turned over. And now let me say something maybe even more dramatic. The proteins, you mentioned proteins, the proteins in the cell are continually turning over too, because you have to have the right protein in the right place at the right time. And there is this mechanism by which proteins can be degraded, and the cell expends quite a lot of energy in continually degrading and reconstituting those proteins, because the integrity of the proteins have to be just right, otherwise life doesn't function well. So you have here everything turning over all the time, but the point is, and this is the major issue, this is the big discovery of that dynamic kinetic state, is that chemistry, the chemistry of biology isn't the regular stuff that chemists do in the lab, adding this to that to get something else. It's creating this fountain, this chemical fountain at many, many levels for its functionality and its fantastic capabilities, including, and we'll get to that, I hope, eventually, thought, mind, and consciousness.
So why does it sometimes go, but we will get there, definitely. And so I think, you know, I find it a compelling argument that certainly, you know, things are turning over within each of our cells all of the time. There's no doubt about that. That's absolutely right. I like the argument about this dynamic kinetic state, and I like the arguments that once that gets going, you can end up to get to a sort of more complex structure, and it allows you to grow longer reaction chains and stability emerges and what have you. Love that argument. But why does it go wrong, and why do we die?
Because it doesn't go wrong. Nature is very clever. Nature discovers that as individuals, we only, if we're talking now about, let's say, human society, once your kids are out of the house and raising their kids, you're in the way. You are a burden. You're using valuable resources, and nature decides to eliminate you. That's the bottom line. So this is very important. People think that we age and die because we wear out like a used car. That's absolutely not the case. The body is very clever. It knows how to make us stronger and fitter from age 0 to age 20, and then from 20 to 50, it maintains a plateau, very comfortable with that. And then at some point it says, you know what, for persistence, the persistence isn't individual persistent. It doesn't care about us as individuals, just like that drop of water in the water fountain comes and goes. What's important is the stability of the fountain, and the stability of life is a population stability. It's not an individual stability.
So continue. Oh, sorry, Simon, I was going to let you come in, or do you want me to keep going?
No, I guess I was wondering, in that case, why is there such a vast span of ages within the human population? You'd think that maybe, or do you think in the future we may get to a point where we all hit about a certain age and then the universe decides, that's it, and, it all becomes tightly regulated? Or will there always be this distribution of ages?
I'm not sure that I fully understand. Why you're saying, why do we're born at age 0 and then become older?
If the system is working perfectly efficiently, then
Yes.
as soon as we're no longer needed, we would expire.
Yes.
But some people persist and linger on for much longer than others. So does that mean that that is not so tightly regulated as it could be, do you think?
Well, I mean, I'm not sure that I accept that, in the sense that I think the typical age where we die is somewhere, between, let's say, 70, 80, 90, 100. So there is that there's that window where roughly that in the big scheme of things that nature has decided that's where it's time to move on. Clearly, every individual he has to deal with all the contingencies of life independently. So one might get sick with some disease, one might get run over by a motor car, etc. But we're designed, if I can use that word, dangerous word to use there, to last around that 80, 90 type age frame.
So let's come on to consciousness then. So the hard problem of consciousness can be termed in the fact that why aren't there zombies around that do everything that we do but have no experience of it? They don't experience the world.
Yes.
Does this kinetic dynamic state that life is in, where we're a flow of energy through, we're continually turning over, we're doing work, all of this sort of stuff. Does that logic find a way around, say, the hard problem of consciousness? Why are we aware? Why are we sentient?
Yeah, no, that's a very good question. And I felt that a couple of years ago, when I suddenly realized that the DKS state opens the door to mind, And that's the basis for the new book that I'm now writing, which is the physical mystery of life, the road from matter to mind. That's the title of the book. It opens up a means of understanding where mind came from. Now, you see, I have to go back a step here. Modern biology has, I mean, I think biologists suffer a little bit from what one might call physics envy. They wanted biology to be very physical, and there was an excessive emphasis on physicalizing biology for much of that 20th century, trying to see biology as a branch of physics. But that was a problem because physics hasn't been able to tell us what is mind and what is consciousness. So basically, they were latching onto a physics that wasn't delivering the goods. Now, and there is this enormous gap still between the physical and the biological sciences, and I feel we're getting closer to bridging that gap. Now, what's the answer here? And you see, it's interesting. One very clever woman, Barbara McClintock, already many, many years ago, already sensed that this idea of just, it's all like physical, mechanical, is wrong. She was already aware that that life, living things have an awareness. She was aware of that, and she even spoke about that in her Nobel Prize speech in 1983, which was given to her some 40, 50 years after she made her big discoveries, because people basically said, that can't be right, what you're saying, that genes, move around, jumping genes, and that she was the first to realize that there is a dynamic character to life, and it's already present within the genes. So a genome is not this static place where information is stored. It is part of the dynamic process of life. And the genome is also in that dynamic kinetic state, because if it isn't, it's going to be very hard to access the bits of it that you need at any given moment. So, but now coming to awareness. Look, this is a tricky thing to say, but that water fountain, in some sense, is aware of its environment. It's aware of its environment in the sense that if the water is cut off or the energy is cut off, it ceases to exist. So it is existentially dependent on its environment, and that means there's an awareness there. Now, in a physical system, that awareness means nothing. Obviously, I'm not saying water fountains are aware of their environment, but once you have a chemical system in that DKS state, and it can undergo modifications, then it modifies itself to become more aware of its environment in order to move towards increasing persistence. In other words, there is a directive, because the evolutionary direction is towards greater stability, and in fact, that contradicts one of the tenets of modern biology, that evolution has no direction. It does have a direction. It's direction towards increasing stability / persistence. Okay, so there is the beginning of awareness, and that awareness, if you like, is the beginning of mind. So mind started the moment a DKS system emerged. Mind started in chemistry, and incidentally, Charles Darwin said the evolutionary process moves along both physical and mental axes, not just the physical one. For most of toward the end of the 20th century, the view in neurobiology is that mind is something, nervous systems, brains, etc. This is a later acquisition. But Darwin understood something, that you can't just suddenly, in the middle of an evolutionary process, go off on a tangent and discover mind. It has to have a beginning. Evolution improves things. So what I'm saying is that that first chemical system had the beginning of mind because it was, if you like, aware, in inverted commas, of its environment.
Well, so it could sense it could sense aspects of its environment,
Yes.
and it could respond to it. I don't think you're really saying it's sentient in a sense, but you're saying there is or are you? Are you saying, that even that very, very first protocell, that collection of chemicals that got into this dynamic state, had some degree of self-awareness?
It is inherently, you see, if it's aware of its environment, then in a sense, it's aware of itself. Its dependence makes it, there's the beginning of self-awareness through external awareness. And this, because living things, of course, continually want to improve themselves. So that's part of the evolutionary process. So the evolutionary process involves changing your environment so that you become more stable, more persistent, and changing yourself to become more stable and more persistent. In other words, the two things go hand in hand. And that means when David Chalmers asks, why don't we have zombies around? Why do we need that feeling of awareness, sentience? Because without that, it's not alive. You see, if you make a robot that does all the things that we can do, it's not alive. It might be an excellent robot. It might make you a cup of tea or whatever, but it's not alive. It's not aware. That awareness comes about through this dynamic state, and therefore that it's there from. Without that, you don't have life.
So, Addy, does that leave things like viruses in a peculiar place? Because obviously, we don't consider viruses to be living, but they are aware of their environment.
Okay, viruses is an interesting situation in the sense that they are not alive in this DKS sense, because they are just, if you like, a static aggregate.
Yes.
But an unusual one that when it's placed in a replicating environment, the cell by mistake starts making copies of it. So it is something that is like is hijacking the life system. So it's not alive and it doesn't have a sense of any self-awareness. It's just it's a non-living thing. But life, and this is very important, and it comes up when you mention viruses, life is a network. It's a network that continues to grow and expand. So initially, the network was chemical. It then became cellular. Then it goes to the multicellular level where cells are making a network with all the other cells in our body. And then we make a network with other people. What we're doing now is networking. And of course, it's not just humans networking with themselves, but our biome, where the network includes bacteria and viruses, and we're all one huge, I almost said happy family, not always so happy, but we're one big family. And interestingly, the attitude to Gaia has changed. When Lovelock initially raised that idea, it was considered, capability. This is like one giant organism. Slowly, biologists are softening with regard to their rejection of that idea and beginning to see that the entire life network on this planet is an integrated one where everything is connected with everything else because it increases stability, persistence. If you have this very persistent system, very complex and persistent system, then if something terrible happens to a part of it in one place, it won't affect the survivability of the system as a whole, because it's got all of this complexity that ensures that the system can bounce back.
So I think where do you go next? So I think there's some really compelling arguments here. I like the idea about the kinetic dynamic state. I think there's no doubt that things turn over. I'm yet to be convinced about some of the consciousness arguments, if the truth be told. But that's how science progresses. People put out ideas, we discuss them. So it's really exciting. But where next then? What are the next steps on the what are the key killer experiments that people are doing at the moment, or the key kind of killer bits of theory that have to be developed for these ideas to become universally accepted by biologists, physicists, and chemists, and for physicists to end up with biology envy?
Well, the great Richard Feynman once said, if you don't know how to make it, you don't understand it. And what I'm trying to show in a very rough way, in a very general way, is what are the outlines of a system, a chemical system that could evolve, that one could make and would evolve in the evolutionary direction to move towards life. So basically, and this is something that I mentioned him a little earlier, Sibrin Otto is trying to make chemical systems that are in this DKS, replicative chemical systems in the DKS state. And that is a big ask. And that's the challenge. And the guy that can do that, I think, is in line for the big one, as one might say. But that is the big challenge. But there is at least a goal. I'm saying that there's a way, a theory is of no use if you can't test it, if you can't substantiate it. So that would be the test, to create a replicative system, not a molecule on its own. It has to be in the dynamic state, which is able to replicate, and also it has to be structured in such a way that its evolutionary direction is open-ended. In other words, there's a direction by which it could evolve. And of course, the sequences of nucleic acids is ideal for that. And that's why that's such a central part of the evolutionary process.
Simon, any kind of final thoughts? Just keeping an eye on the time here. This has been fascinating. I'm really enjoying it, I have to say. And as a scientist, one has to have a bit of skepticism, but I think there's something definitely there. I'm persuaded.
Yeah, me too. I'm still thinking about the conscious thing and the awareness aspects, as you said, Tim. But yeah, I think it's been fascinating, and really looking forward to the new book too.
So yeah, your book is. Your book on how what is life, how chemistry becomes biology. That's been out for a little while. You can buy it, and it's in the Oxford Landmark Science series, if I believe. Is that correct?
Correct. That's right.
Yeah, and it's a really good read. So I really enjoyed it, I have to say, and it's been fascinating talking to you, Addy, and a real pleasure.
Thank you very much. Great talking to you.
Well, that was interesting, I have to say. What were your thoughts, Simon?
Yeah, absolutely. I mean, it's growing up as a chemist, it's another way to think about chemistry, and this sort of balance between thermodynamics and kinetics, and that it might, that interface isn't quite as sharp as we thought it may have been. And there's this state that's sort of kinetic, but in seems more like a thermodynamic state, but actually it's defined by its kinetics, is actually very eye-opening.
So just for our listeners, in case they're not kind of fully up to speed, kinetic means something to do with reactions, is that right? Yeah,
so kinetics means something to do with reactions, it means to do with movement, And it's also about the speed and rates at which things happen. Whereas thermodynamics is more something we consider to do with sort of, we tend to think about something called thermodynamic stability. So we think of things very defined. As we said, they can be solid, they can be liquid, they can be gas. But it now seems that there's states that can be in between that, and that their chemistries are in flux, frankly.
Well, I always thought I was in a bit of an odd state, so I think
You are for sure, Tim.
I mean, I think he's absolutely right that if you think what's going on in the cells in terms of turnover and, yes, a cell's a thing, but it's kind of in flux. There's all this process going on inside of it. So I think that's completely accept that. Less persuaded by the arguments that we could that we don't wear out. I mean, I think there's quite compelling evidence that our mitochondria, for example, so the little powerhouses in our cells, do wear out, and we're not, for whatever reason, we're not able to wholesale replace those.
Right.
And I'm not sure I fully understood or bought the consciousness argument, but I think for life getting going, it's actually, I think it's a really neat idea, a really nice idea that there's this, because I think it's true. It's very hard to see how you can get it going with a lot of the sort of standard chemistry that we understand in the lab. Is that right?
Yeah, I think so. And I think also when it did start, the conditions around us will not have been the conditions that we have now. So it could well be that maybe they did resemble those in a very, very specialized lab in some way, because the pressures were different, the temperatures were different, the environmental composition, the composition of the air was different. It's not quite as it is now. And so the chemistry would have had to have been different. Yeah, I agree with you totally. And the bit about consciousness, yeah, I think so too, because I think simply responding to an environment or is not the same as being aware of an environment to me, I think.
Yeah, I think that's right. I think that it's true that bacteria, for example, can sense chemical gradients and can move away with that, by flapping their cilia, move away or towards that signal. But and that's an impressive thing to do, but I don't think that suggests. I think awareness isn't quite the right word there. I mean, I can see where he's coming from, and maybe that is a first step on the journey, but it's not. Yeah, I think I'd agree with that.
There's still a leap, isn't there, to the yeah.
Anyway, I thought, no, it was fun, and I think it's always really interesting to, we need kind of scientists who are pushing barriers and coming up with new ideas, and his book really is worth a read. So I enjoyed it, and really nice chatting to him. Really good fun.
Yeah, a lovely person and open-minded, and I think I'm naturally drawn to him because that's where my interests are too, at that interface between chemistry and biology and physics. Let's let the physicists in here too.
Oh, do we have to?
You have physics envy after all, Tim.